Phosgene, the spectral killer of World War I. Also known by its military designation as chlorine gas, phosgene gas is a synthetic compound generated by passing carbon monoxide and chlorine gas through a porous bed of activated carbon. Nowadays, it is used in the industrial synthesis of isocyanates. However, during the height of the First World War, phosgene was an airborne toxin released from 50-pound canisters that the British affectionately referred to as mice. Being denser than air, phosgene would sink into the trenches below and wreak devastation on the lungs and the morale of the troops within. Should an unfortunate soul not get his or her gas mask on in time, the small hydrophobic phosgene molecule will enter the lungs. This planar molecule is well within the minuscule nanoscale required to bypass the body's external defenses like the mucus and cilia found within the lungs. Once inside the lungs, the phosgene gas will cross the alveolar membrane via simple diffusion. This is possible due to the small size and neutral nature of the molecule. It is important for respiratory absorption that phosgene can diffuse across the membrane, as there are little to no transporters in the alveolar membrane. It is difficult to assign a drug likeness to the chlorine gas because it is highly reactive with water, making obtaining a partition coefficient impractical. Furthermore, being composed of only 4 atoms, there aren't really any side chains or functional groups to consider. Essentially, this means that at physiological condition, phosgene gas will appear as is, a simple yet deadly compound. Phosgene undergoes absorption for a reason. As a toxin, it is highly capable of damaging the body and the cells within. The focus of this presentation are the toxic effects of phosgene gas on the B lymphocytes in the blood. B cells belong to a group of white blood cells known as lymphocytes. Lymphocytes are an important part of the adaptive immune system that is responsible for recognizing and reacting to foreign material within the blood. These potential pathogens display antigens, little protein tags that protrude from the surface of the cell membrane and identify its origin. Specifically, B cells make antibodies. These Y-shaped structures recognize antigens on a cell's surface and bind to any foreign intruders, acting like homing beacons for other immune cells. After encountering a specific pathogen, some B cells become memory cells. The distribution of phosgene gas is an interesting scenario. The ultimate target of the toxin are the B lymphocytes found from the blood. As such, the moment the phosgene gas crosses the alveolar membrane, it has reached its destination. In fact, small hydrophobic molecules like phosgene are absorbed in the bloodstream within seconds after inhalation. In general, there are certain benefits of being absorbed to the respiratory system. The most important aspect is that the toxin skips first-pass metabolism. Normally, toxins ingested orally will cross the gastrointestinal tract and are shuffled into the liver to undergo metabolism. Although this is unlikely to be the case for phosgene, which is rarely a solid, as it boils around 7 degrees Celsius, it is worth noting as one of the advantages of being absorbed to the respiratory system, specifically to this toxin. In fact, the major dilemma for distribution of phosgene into the blood is its reactivity with water. The humid environment within the lungs means that phosgene is susceptible to hydrolysis before it even crosses the alveolar membrane. If this is the case, the resulting carbon dioxide and HCl produced is unable to work its effects on the B lymphocytes. Metabolism refers to the biotransformations of a toxin, like phosgene gas, by the chemical processes within the body. Depending on what the toxin is, different forms of metabolism will occur. Phosgene gas undergoes 2 forms of metabolism, Phase 1 and Phase 2. Phase 1 metabolism consists of 3 reactions: hydrolysis, reduction, and oxidation. The primary role of these metabolic processes is usually to expose a functional group like a hydroxyl or an amine. However, due to the small number of atoms in the phosgene gas, there aren't really any functional groups to expose. Instead, hydrolysis of phosgene gas is one of the main ways of removing the parent toxin. The highly reactive nature of the chlorine species, these metabolic reactions that normally require catalytic intervention, can occur without an enzyme. Hydrolysis comes from the Greek word hydro, which means water, and lysis, which means separation. Therefore, as you would expect, it involves the breakdown of a toxin with water. This occurs primarily due to the electrophilic nature of the phosgene gas. As seen in the top panel, a water molecule acts as a nucleophile, attacking the phosgene gas and releasing chlorine. Being a stable leaving group, this is possible and occurs during an SN2 reaction. Subsequently, another water molecule acts as a nucleophile and removes the hydrogen from the hydronium group on the phosgene gas, producing a carboxylic acid and hydrochloric acid. Focusing on the bottom panels, we see that as a stable leaving group, chlorine can dissociate from the carboxylic acid, and is usually by the formation of a double bond between the second oxygen and the carbon. This is brought on by the nucleophilic nature of the lone pair on the oxygen, which attacks the electrophilic carbon center, which had been stripped of all its electrons from the highly electronegative chlorine atom. Ultimately, what is produced is carbon dioxide and hydrochloric acid. Where hydrolysis occurs has different effects on the absorption and excretion. If hydrolysis occurs on the humid surface of the lungs, the products are carbon dioxide and hydrochloric acid. The CO2 is simply exhaled and the hydrochloric acid irritates the alveolar wall before being neutralized. This means that absorption to the bloodstream does not occur and excretion simply consists of exhalation. If hydrolysis were to occur in the blood, a similar process happens. CO2 still readily crosses the alveolar membrane into the lungs because it is relatively high concentration within the blood. The hydrochloric acid, however, is instead neutralized by the bicarbonate buffer system and easily managed. The bicarbonate buffer system is a dynamic equilibrium between carbon dioxide in the blood and carbonic acid. The interconversion of these two forms is supervised and overseen by the enzyme carbonic anhydrase. Phase 2 metabolism almost always follows Phase 1 metabolism. However, it is possible for the two events to be separate, and it is the case for phosgene gas. Phase 2 metabolism consists of glucuronidation, sulfonation, acetylation, methylation, conjugation with glutathione, and conjugation with amino acids. Phosgene gas is involved in acetylation. As the name implies, acetylation is the addition of a acetyl group to a toxin. This renders it more hydrophilic and soluble in urine. An acetyl group normally includes a carbon, double bonded to an oxygen, and single bonded to a methane. The fourth carbon bond varies between acetyl groups. In this special case, the methane is replaced by a chlorine atom. Acetylation usually occurs to amine groups, sulfide groups, and hydroxyl groups. The lone pair of electrons present in these groups act as nucleophiles that attack the electrophilic carbon center of the phosgene gas. As a result, the chlorides, which are great leaving groups, dissociate and form the familiar hydrochloric acid seen as a byproduct of Phase 1 metabolism. This HCl is buffered by the bicarbonate buffer system and is only dangerous at high concentrations. The resulting acetyl products, however, must be further oxidized by acetyl hydrogenase in order to become carboxylic acids that are easily excreted into the urine. Phosgene gas presents a threat to B lymphocytes both directly and indirectly. Although there is no consensus as of the exact localization of the action of phosgene, its reactive nature allows it to interfere with several important functions of the B lymphocytes. In particular, B lymphocytes are well known for their B cell receptors. These receptors are exposed transmembrane proteins located on the outside surface of the cell. Although their exact composition varies from antibody to antibody, the general structure contains several targetable groups that include, but are not limited to, sulfide groups, amine groups, and hydroxyl groups. These groups undergo a similar acetylation reaction with phosgene gas and denature the protein. As such, this allows the phosgene gas to be very flexible in what it can inhibit. Phosgene gas can also react with polypeptides and phospholipid heads. These are important because they are part of the cell membrane, and if they are denatured by phosgene, the entire protein, or the entire cell, of the B lymphocytes will be denatured. What makes phosgene gas so dangerous is that its reactivity is not limited just to B lymphocytes. It is well known for its toxic effects on the alveolus. By destroying and degrading this membrane, the B lymphocytes are exposed to the external environment as blood rushes into the lungs. In addition to being extremely painful, this exposes lymphocytes to several potential foreign reactants that can cause damage. Widely used as a chemical weapon, it should come as no surprise that phosgene gas has a devastating toxic effect. Surprisingly, only 2% of the casualties sustained from phosgene gas were fatal. Instead, at the average chemical dose of 4 parts per million, you can not only smell the hay-like scent of phosgene gas, but you experience the degradation of your alveolar wall. Although non-fatal, the phosgene indiscriminately destroys your lung tissue, causing severe discomfort, but also weakens your B lymphocytes and inhibits your ability to combat the subsequent infection that usually follows in the lungs. As such, it is for good reason that these chemicals are highly controlled today in industrial use, as terrorists could easily access these chemicals and cause wide-scale panic. What has defined phosgene as such an effective military weapon was not its physical damage, but the psychological aspect that it caused to the troops. Such damage cannot be measured through toxicology alone and have to be considered when understanding the dangerous implications of this technology.
In conclusion, it is up to politicians to develop strict laws and control of this deadly acetylating gas to ensure the safety of individuals worldwide.